Should I start, Umesh? Umesh, should I start?
Yes, Silpi, ma'am, we can start now.
Yes, you can start, Shilpi.
Very good afternoon to one and all. Well, after the successful accomplishment of two very interesting lectures of Technical Session 4, I, Dr. Shilpi Bhatnagar, welcome all the students, participants, faculty members, and research scholars from India and abroad for the last technical session of the 2 days International E-Conference, followed by the valedictory session. Now, I would like to welcome and give a brief introduction of the chairpersons of this session, Professor Ruby Mishra, Department of Chemistry, Deshbandhu College, University of Delhi, and Dr. Roshni Rajamohan, Department of Botany, Deshbandhu College, University of Delhi. Professor Ruby has more than 15 years of teaching experience. She is working as NSS Program Officer in Deshbandhu College. Under her supervision, she has taken various issues on NEP, Mission Kavach during COVID times, and Mission Kalindi to clean Yamuna River. She has been associated with various professional bodies such as life member of Vigyan Bharati and Indian Society of Analytical Scientists, Delhi chapter. Dr. Roshni Rajamohan Mathur is an associate professor from the Department of Botany, Deshbandhu College, Delhi University. She has a teaching experience of more than 25 years in the UG and PG courses. She is involved in active research and is a recognized research supervisor, guiding students in the field of plant systematics, physiology, and medicinal botany. She is an elected fellow of the Society of Plant Reproductive Biologists and life member of various professional bodies. It is my high privilege to welcome Professor Devan S. Rawat, FNASC, Dean, Examination, University of Delhi. Now I invite Dr. Roshni Rajamohan to give a brief introduction of Rawat sir, our keynote speaker of today's session. Over to you, Roshni ma'am. Has Roshni ma'am joined, ma'am? Ma'am is joining in a minute. She's busy in college with another research session.
लीना रोशनी मैम को को-होस्ट बनाइए.
Sir, by what name she is joining?
रोशनी के नाम से ही करेंगे लीना.
She is the co-host now.
Yes, Roshni मैम, please.
Hello.
Yeah. So you have to introduce Professor Rawat, sir.
Yes. I have spoken to Shilpi.
You can start.
Is it a call for me?
Yes, ma'am. Yes, ma'am.
Yes, ma'am. Yes, ma'am. You have to start.
Thank you, Shilpi. Very good afternoon to all those who are assembled here. So this two-day international e-conference on recent advancements in society, organized by Department of Chemistry, Deshbandhu College, University of Delhi. At the outset, I would like to thank the organizing team for giving me this opportunity to chair this post-lunch session on day 2 of the event. I feel privileged and delighted to welcome and introduce the Honorable Keynote Speaker for the day, Professor Diwan S. Rawat. Professor Rawat is an outstanding teacher of the Department of Chemistry and Dean of Examinations, University of Delhi. He is also a visiting professor at the Japan Advanced Institute of Science and Technology, Japan since 2016. Dr. Rawat began his academic journey from Kumaon University, Uttarakhand, and went on to complete his PhD from CDRI, Lucknow. He has 2 postdoctoral fellowships in medicinal chemistry from Indiana University and Purdue University, USA, and he joined University of Delhi in the year 2003. Professor Rawat has served as an associate editor and has been a member of International Editorial Board in various internationally reputed journals like Nature Scientific Reports, RSC Advances, ACS Bioconjugate Chemistry, Journal of Biochemical and Molecular Toxicology, Marine Drugs, and many more. He has part of many research assignments at various institutes across the world. And has rendered his services as an I think I got disconnected in between. Am I audible?
Yes, ma'am. Now you are audible. You can continue.
I'll just continue. Sorry for the disturbance. Okay, I'll just continue introducing Professor Rawat's academic achievements. Professor Rawat, for his contributions in various fields like academics, research, and administration, has been well acknowledged and is very much evident from the impressive list of awards and honors he has received. He is Fellow of National Academy of Sciences, Allahabad, is a recipient of Vasvik Research Award. He's also received Special Appreciation Certificate for Exemplary Services from University of Delhi in 2021, Pratap Bhaiya Smriti Samman, Acharya Narendra Dev Siksha Sansthan. He has given the Platinum Jubilee Lecture at the Indian Science Congress and many more honors. It's a long list which goes to his credit. Above and all, Professor Rawat, in his illustrious career, has an envious number of 158 publications in internationally reputed journals of high impact, with over 5,915 citations. He has an H-index value of 45, ITIN index value of 123, with 7 patents to his credit, a book and 4 book chapters. Such a person who is a leading example for all of us. I now invite Professor D.S. Rawat to deliver his keynote lecture on the topic, Journey from Anti-Malarial Molecule to Clinical Candidate for Parkinson Treatment, which is based on his latest internationally acclaimed work. Before Sir can start, I request all the participants to post their queries in the chat box, which will be taken up at the end of the lecture. I invite now you, sir, to deliver your lecture. Professor Diwan D.S. Rawat. The floor is all yours, sir.
Sir, please unmute yourself.
Yeah, sorry. Thank you so much for your kind word of introduction. My thanks to Dr. Umesh Kumar and Dr. Rajiv Agrawal for the invitation. Am I audible?
Yes, sir.
Okay. So today I'm going to share some of our work which we started after joining Delhi University way back in 2003. Let me congratulate the department and the college for organizing such a wonderful conference and inviting some of the most well-established chemists of the country and abroad. My congratulations to you. Before going to the technical part of my presentation, let me just share what the drug discovery means actually. Okay. What was the drug discovery? I mean, how the treatment of the alignment of the people used to be there before the birth of organic chemistry. So until the middle 19th century, the nature's pharmaceuticals, what we call natural products, were used to be used for the treatment of the pain and suffering of the human beings. The oldest evidence of medicinal plants use for the preparation of the drug has been found in the Sumerian clay slabs from Nagpur in India, that is about 5,000 years ago. And this is the Sumerian clay slab that was found in Nagpur. This comprises 12 recipes for the drug preparation, referring over 250 various plants, some of them alkaloids such as poppy, henbane, and mandarin. So this was the oldest example of uses of medicinal plants for the treatment of various kinds of diseases. That was found in Nagpur about 5,000 years ago. And you know, Indian Vedas talks about the healings by the use of different kind of formulas. The oldest one is Shustra Samhita, as you know. This is about 1000 to 500 BC old, and it is one of the fundamental textbook of Ayurveda, or the Indian traditional medicine, what you call. The surviving text contains a description of over 1100 illnesses, 700 medicinal plants, their safety, efficacy, doses, and benefits. This is what it is listed about 1000 BC. You know, and the FDA came in existence late in 1940s when the thalidomide disaster, around 60s when the thalidomide disaster occurs. But it was already there in our system. When you want to sell something, use something for the human use, you need to have some kind of safety precautions, and efficacy and doses, and what will be the benefits or what can be the disadvantages of taking those kind of formulas. But when we come to the synthetic drugs, the oldest example of the first organic compound was urea, that was synthesized by Euler in 1828. Let me tell you, Euler was not a chemist actually, he was a medic. And he was taking his subsidiary classes of chemistry with Professor Gamlin. And Gamlin realized his potential as a chemistry student, and he advised him to take the classes of J.J. Berzelius. J.J. Berzelius was the chemist who proposed the hypothesis, what it is called vitalis. Vitalis means, according to his hypothesis, organic compounds can be synthesized only in the living system. So Euler did PhD with J.J. Berzelius and Gamlin and published his first research paper that talks about synthesis of organic compound in the laboratory. So that was the first compound synthesized in the laboratory in 1828. The first synthetic drug is a coral hydrate that was discovered in 1869, and it was introduced as a sedative hypotonic, and it is still used in some part of the world. And the first analgesic and antipyretic like phenestin, acetonolide were the simple chemical derivatives of aniline and paranitrophenol, and both of which are the byproduct of coal tar. So that comes from the nature. And one of the oldest example of nature's pharmaceutical is the willow bark. Okay, this willow bark was used for the treatment of pains and fever for centuries. And later on, a compound that was isolated is called salicin, and that salicin was nothing but acetyl acetic acid. And that was, the aspirin was synthesized in 1897 by Hoffman. So from there, the role of organic chemistry started emerging, and how it impacted the human health. Let me show you in the next slide. This is what the life expectancy at birth in 1900. Look at India. The life expectancy in 1900 in India was about 23 to 24 years. In 1950s, it goes about 37, 38. Today, we are around 65, and Japan has the highest life expectancy. How that happened? This is because the chemistry played a pivotal role in the development of the drugs. Because earlier days, though natural products were being used as a crude mixture for the treatment of different kind of sicknesses, which may not be that efficacious. So when we talk about medicinal chemistry or drug development program, it is kind of a two-part you can divide it. One, what we call a rational drug design, and second part, what we call drug design or repurposing drug. Repurposing drug came in a very big way actually during this COVID time, because drug discovery program, I will show you in the next slide how tedious it is. It is not that easy. But when we say repurposing, what does it mean? It means you have a known drug, you use this drug for the treatment of some disease. Let us see if the same drug can be used for the treatment of some other disease. If that can happen, you don't need to worry about so many other parameters which delays the drug discovery program. So when we say rational drug discovery program, what does it mean? It means the program starts from the chemist actually. This is what the student need to understand why chemistry is so important. There is one branch of the science which never can die. That is the chemistry, especially the organic chemistry, because organic chemist is the person who makes things, who makes the carbon-carbon bond. So we are an architect. So if you have that kind of knack to making something, you cannot be unemployed. So this is how the medicinal chemistry goes. So medicinal chemist makes compound in the lab. The synthesis of those compounds has some kind of rationale based on your knowledge about the particular kind of disease. And then you synthesize the compounds and characterize them. Then you do the in vitro or in vivo screening. If that goes well, then you identify the hit molecule. That hit molecule is optimized. Then you try to find out the patents at that particular stage of your work. Once you have a patent, then go for preclinical, then clinical development, then registration, and go for marketing. The other way is you go for in silico design, means you first do the molecular modeling and other kind of stuffs, try to find out what kind of molecule can act against this particular target. Once you have that studies with you, then go back to your laboratory in the chemistry laboratory, make those compounds. Then test the hypothesis in the biological system, whether that works or not. Having said that, when we look at this slide, it looks not that difficult. It looks a bit easier though. Let me show you another slide. This is how the drug discovery program goes actually. So when an organic chemist synthesizes about 10,000 compounds, one compound comes to the market as a drug, number one. Number two, at different phases, your molecule will get out from the studies because of one reason or the other. Let me show you how much this cost. It cost about 460 million US dollars. It's the average cost of drug discovery. So you can think it takes about 15 to 16 years, number one. Number two, 460 million US dollars. So why I show this slide? Because wherever I go, I show this slide first, so that at the end of my talks, nobody asks me question, have you developed any drug molecule? So this is to safeguard that kind of question at the end of the talk. Okay. So when we say drug discovery, actually, when we work on this area, there are three different approaches. One is rational drug design, then target-based drug design. The third one is the repurposed drug design. The repurposed drug design, those who watches the news daily will have understood or heard about it during COVID period, when HCQ, hydroxychloroquine, was initially used to combat this COVID menace. The hydroxychloroquine is nothing but a drug that was used for the treatment of malaria way back in 1960s and 70s. Still it is used in some cases. So this was the drug already available in the market, used. So its efficacy and this clinical trial and all that is already known. So it was thought to use this for the COVID virus, and it was used many places at that time in the beginning. So when I say the drug design, let me just talk to you about one approach, that what we call the hybrid drug design concept. So what this hybrid drug design concept is? It's very simple when you explain it. Now, just think about it. If you are sick and you visit a doctor, okay, and most of the time what happens, doctor gives you more than one medicine to treat one particular disease. So it means this particular disease, what the doctors has investigated, cannot be treated by a single agent. The most interesting example of such kind is the tuberculosis. Tuberculosis cannot be treated by a single drug. You need to have a multi-drug treatment. Now, when we say, let us suppose you take two drug molecules to treat some of your sicknesses. When you take the molecule, it goes into your body. Just think about it. What might it happen? Because your liver has high acidic pH. So what happens? The molecule, when it goes to the biological system, get metabolized. So T half of the molecule determines the activity or the safety profile of the molecule. So it means if you are taking two drugs, the T half of both the drugs cannot be identical. Number two, they can interact with each other. So now as a chemist, you can think, let us suppose you have drug A, you have drug B, hook up these two drugs by a covalent bond. This is what we do as a chemist. We make a carbon-carbon bond. So I had a drug A and I have a drug B. What I did, I put a covalent bond in between. So when I do that, what happens? I have one molecule now. I don't have a two molecules. It's a one molecule. But this one molecule has both the pharmacophores, both the drug molecules. And let us see if that works or not. This is what the concept, what we call hybrid drug design. It is not old concept actually. It was, the first example was reported in 1987 or 88. So we started working on this concept. Let me just show you what I said. This is the typical polypharmacology method of treatment. You have two tablets. Two tablets mean two compounds, and you treat your sickness. That's one. Another one is you have a capsule. And those who have seen the capsule have different colors also, right? So what does it mean? It means this capsule has two different organic compounds mixed. It has solved the problem to a larger extent, but the problem, there are different problems. As I said, the difference in the relative rate of metabolism between the patient. So the rate of metabolism of both the drugs will be different, but this will be different to patient also, because it depends on the enzyme activity of one patient to another patient. So to correlate those data is not that easy. And that gives you a complex metabolite at different rates, leading unpredictable variability between the patients. This is one of the issues. But when we say hybrid, this is now hybrid. What it means this compound is one, another compound is this. These are covalently bonded each other. So if that works, it can have a better patient's compliance. Okay. Single molecule, so lower drug-drug interaction, better and predictable pharmacokinetic profiles, structurally different compound, so drug resistance can be minimized. So these can be the advantages if this kind of approach works. Now let me just show you. This is the first example of hybrid drug design. It was published in 1997. If you look at these two molecules, this is called elliptixin and this is epotoside. And this is the compound which is a mixture of these two. What is the mixture? This part of the molecule, this part, can you see the cursor? Cursor dikhai de raha na?
Yes, sir, it is visible.
So this part of the molecule is here. And this part of the molecule is here. Look at this. This is what it is. So this molecule is a hybrid of this part of this molecule and this part of this molecule. And when studies were confirmed, and both of these compounds were anti-cancer, both of them had different target individually, but when they were hybridized like this, they showed a single target. And activity was much better than what is reported. Let me show you a next slide. This is what experimentally it was proved. But nature has already given us those kind of molecules which we never anticipated or never studied those. Let me just show you the next slide. Look at this. I hope you all have seen this plant, what we call Sadabahar. Small plant. You know, this plant has been a source of 70 alkaloids. 70 alkaloids have been isolated from this plant. Out of these 70 alkaloids, I have listed 2 alkaloids here. These 2 alkaloids were isolated from this plant, and both of these compounds showed poor anti-cancer activity. But the same plant gave 2 another alkaloids. What were those? This one. When these are covalently bonded like this is called vincristine. And if you have a methyl group in presence of the formyl group here in place of the formyl group, that is called vinblastine. Vincristine and vinblastine is the first drug to be licensed by FDA in 1963 for the treatment of leukemia. This is what nature has given us. We never analyzed that. 1963. Now 1997, a designed molecule based on this particular concept, which nature has utilized, proved yes, it works. The next example is of that kind of hybrid molecule. You know, this is a quinine, old age molecule that was isolated from cinchona bug and used to be used for the treatment of malaria and high fever. Later on, in 1930s, 1940s, this molecule was synthesized, what is called chloroquine, again a drug for the use for the treatment of malaria. And this is the molecule which was isolated by Chinese group in 1969 or 70. And this molecule is called artemisinin. This is another molecule used for the treatment of malaria, especially when you are infected with Plasmodium falciparum. If you are infected with Plasmodium falciparum, chloroquine doesn't act. It's not useful. So you used to have this molecule. Recently, not recently, maybe around 10 years back, this molecule was synthesized by erstwhile Ren vaccine, and it was licensed for the treatment of malaria. This is a hybrid of this trioxane, which comes from artemisinin, and this part of the molecule comes from some other drug molecule. And this is the lady who got the Nobel Prize for the discovery of artemisinin in 2015. So based on this, what we thought to make hybrid molecules based on, this is the chloroquine, we thought take this aminoquinoline part from chloroquine, and these were the other two drug molecules used for the treatment of malaria way back in 1960s and 70s. So we thought take this red part of this molecule and hybridize it and make the hybrid molecules. Now, chloroquine stops the hemojoin formation. The mode of action is this. But these two classes of compounds, this is cycloguanine and trimethamine, they stop the DHFR inhibitor. They were the DHFR inhibitor. So both of them are anti-malarial, but their target is different. So what we designed? We designed something like this. So this red part of this molecule comes from chloroquine, and the blue part comes from the trimethamine, and you have a linker here. Another one is we have a PFCRT enzyme inhibitor, that is another class of compound, quinoxylin based compound. We thought to design these kind of molecules. So we did that. Let me just summarize the first part of our work that was done during 2006 to 2009. What we did here, if you look at this part comes from the chloroquine, and this is the other part that is a 5-member with 3 nitrogens. So this is the slide, what it shows the anti-malarial activity of those compounds. This is the IC50 value of chloroquine, that is 0.42 micromolar. But if you see the height of the bar of our compound, it is higher than chloroquine. It means this kind of hybrid is not active. Next one, look at what is the difference now. We kept this triazole, but we introduced one more pharmacophore. That's the triazine that comes from our previous slide. Now look at chloroquine is this, but the bar of the height comes down. So it means these kind of hybrids have shown anti-malarial activity. Go to the next one. Now we remove this triazine, this triazole. We kept the triazine here. Look at this. Now look at the IC50 value of the compound. This is chloroquine, this one. So now you see these bars, the bars which are smaller in height, in comparison to the chloroquine, that is last bar, it means the compounds are better than chloroquine. So what we proved here, if you hybridize two different pharmacophores, you can improve the anti-malarial activity of those kind of hybrids. As a medicinal chemist, if you have these kind of data, you would like to move further. What is that? In medicinal chemistry, what we call one change at a time in a molecule. So you cannot make two changes in a molecule because you won't be able to make a story out of it. So what we did, just do that. Remove one nitrogen by CH. That would become pyrimidine now. Let us see what happens with that molecule. Whether the activity retains number one, or it drops, or it improves. These are the three questions which we wanted to address. So what we did, we used this 4,7-dichloroquinoline, did the aromatic nucleophilic substitution reaction. This is where the chemistry knowledge comes in picture. Fourth Cl get displaced, not the seventh. So this is why the heterocyclic chemistry is important. So a student should read those concepts. So if you know that, you can answer it. If you don't know that, you won't be able to answer. You can substitute both the places. That is not going to happen. So then once you have this, then we took this kind of dichloropyrimidine. You have two regioisomers. One of the Cl was substituted by carbocyclic amine. We got these kind of molecule, and this was characterized by X-ray crystallography. So we confirm which of the regioisomer is which one. Once you have that series of compound, you can't, in medicinal chemistry, you have to make large number of compound structurally similar. Small changes, gradual changes. Then get the biological data done. Analyze those data in terms of the structure. This is what we call structure activity relationship studies. So this was our previous compound. What is the difference now? One nitrogen is less. That's it. Okay. Now, when we did the in vitro anti-malarial activity studies on those compounds, just look at the compound, this compound versus this compound. What is the difference? Look at the attachment here. In this case, the nitrogen carbon bond is in between two nitrogens. In this case, you have a nitrogen here, you have a Cl, you have a carbon, you have a CH. So these are the two regioisomers. Now, if you look at the anti-malarial activity of both these regioisomers, look at it here, it is almost identical, 0.21, 0.27 or 0.17. So what it tells you the point of attachment of the aminoquinoline with pyrimidine doesn't affect the anti-malarial activity of those compounds. So it means you had one minor product, another was a major product. So you can use the major product to make your compound and study them. But if you substitute this Cl by the cyclic amine, look at the activity now, 0.212, 0.026, tenfold improve in the activity. When you substitute this by morpholine, the activity remains same. But if you substitute the same with N-methylpiperazine and N-ethylpiperazine, the activity goes down to 0.007 micromolar, 7 nanomolar. So any compound, if you get in medicinal chemistry nanomolar range inhibition, you would think that you got a landmine, gold mine. But you need to do two things. Number one, what about its toxicity to the healthy cells? So our study says those compounds are non-toxic. The data what I have shown here is in vitro data. So in vitro data means it is not in animals. So animal can behave in a different ways. So you will be asked to show in vivo data. So in vivo activity it was done. So what we found, 30 milligram per kg body weight for 3 days, 80% cure of the animal. So we published this way back in 2012. Once you have this kind of activity pattern, so you would like to optimize it now. This was the beginning, 2.45 micromolar. This is the product what we got now, 7 nanomolar. And this is the possible modification can be done on this lead molecule, different kind. I will show you a couple of the changes what we made. So the first change, if you look at it here, this NH was functionalized. Chloroquine mein dekhiye aap, if you see chloroquine, the terminal nitrogen has diethyl. In this case, we have NH. This NH was substituted by these kind of groups, both the regioisomers. And the activity pattern is like this. What it says the structure activity relationship says N-thiophenyl, N-benzyl, N-furanil, and piperonil. This is the order of reactivity. You can improve the reactivity, but this is what the order is. The next change was to change this linker. So what we did, look at it, the piperazine linker, wherein you have a pyrimidine and aminocillin joined by a rigid linker, that is piperazine. Piperazine is rigid actually, okay? Now this is very flexible. Another series was like this, flexibility was kept, but piperazine is introduced. What happens with activity wise? This was the starting. Activity drops when you use the piperazine linker. But if you use this kind of linker, activity improves. What about in vivo? 33.3 milligram per kg body weight, 80% cure. Good compound. The next design was based on PFCRT enzyme. Okay, this is what the synthesis is, and this is how the compound is characterized. This is 13C NMR spectrum. Activity is 6 to 9 nanomolar. So batteries of compounds were in the range of low nanomolar activity. We did some kind of studies to show the mode of action of those compounds. What it is, this is the absorption maxima of heme. And when we treated our compounds, titrated our compounds with this heme, the absorption maxima drops. It means what? It means our compound has association with heme at different pH. And by Jobs plot, what we determined that the association between our compound and the heme is 1:1. We did some kind of molecular modeling. I will skip this slide, but I will just talk about this for a moment. Look at this. Remember what I said. I said the drug discovery program, it takes 16 to 17 years, 460 million US. And this is the compound which was developed way back in 1960s. This is called amodiaquine for the treatment of malaria. It was introduced in the market, but after some time it was withdrawn. Why? Because it shows liver toxicity. Why it shows the liver toxicity? Look at this. In presence of P450 cytochrome enzyme in your liver, this compound undergoes for metabolism and gives you this kind of hydroquinone system, which shows liver toxicity. If you know the metabolism of paracetamol, which is listed over here, and paracetamol also has liver toxicity. People, when designed this molecule, did not appreciate or looked at the paracetamol toxicity, and they designed and developed the molecule. Later on, it was withdrawn. Now, if you want to make a compound based on this only and avoid the formation of this kind of metabolite, you can have two choices. One, swap the position of OH and N-diethyl. Swap the position. If you do that, this kind of metabolite will not flow. Or substitute this OH by a similar size of atom, that is fluorine. If you do that, you will stop the formation of this metabolite. And that was done. It was demonstrated that this compound was equally active as amide. So what we designed? We designed a similar kind of molecule, taken this part from amide and this part of a molecule from our previous studies. And this is how the synthesis was done. So amide is this part. And what is the difference between amide and the compound what we made? This portion. OH substituted by F and N-diethyl is substituted by pyrimidine. This is the mass spectrum. This is the total NMR. I'm not going to talk about it. What about the activity of those compounds? Some of the 10 compounds, 4 compounds have activity lesser than 10 nanomolar. It was 47 times better than chloroquine in vitro. What about in vivo data? 100% cure. And we studied their metabolic studies. The T half of our compound is 16.7 minutes, while amide was about 5 minutes. So what we proved, we proved that experimentally. This is another series of compound. Now, what is the difference here? Look at it here. You have a fluorobenzene spacer, and here this is not this is a cyclohexyl spacer. What happened? Now the activity improved further 2.2 nanomolar. And these compound, after 60 days, 100% survival, the body weight of the mice increases, completely cured. And this is the slide that shows a small change in our molecule can lead you a different kind of activity pattern. Look at this. This is the starting material, what we used. This one. These positive bars, the upside. This is the cancer growth. Okay, these are the cancer growth. If you can bring that down, it means what we'll say inhibition of cancer. But if those lines goes negative side from 0 to -50, we'll say cancer killing. Look at it. Starting material promotes the cancer. But when this NH2 was substituted by pyrimidine, look at the bar height now, comes down. It started inhibiting the cancer. But when this Cl was substituted by N-methylpiperazine, it not only inhibited, but it started killing the cancer cells. But when this, now in this case, it was a benzene here. Look at it here. Some of the compound inhibit and some of those compound kill the cancer cell. Now when this benzene is substituted by 1,3-cyclohexyldiamine, look at it here. Kills the cancer cell. And this is what the selectivity is. This is the beauty of chemistry. When you play around, then you will come to it. So at that particular point of time, actually, I got an invitation from Professor Kim from Harvard Medical School. He wanted to collaborate for a Parkinson. Why this so? Suddenly, from where this Parkinson come? How much time do I have?
So 10 minutes.
Okay, good. I think I will be done. Okay, so from there we moved to Parkinson. How? This is why it is so important. You should read literature every time. I came across a paper that was published in Nature in 1987, which concludes chloroquine or similar drug could be used to retard the progression of Parkinson's disease in human system. and nobody pursued this. Professor Kim contacted us because he screened 960 FDA approved drugs, got 3 hits. If you look at all these 3 hits, has of aminoquinoline pharmacophore. Look at our compound. Our compound also has this pharmacophore. We started collaborating with him. What is this Parkinson by the way? We have a small part of the brain that what we call substantia nigra here. Okay, and about 10 million people worldwide are affected with this disease, and there is no drug. L-DOPA is given to manage the disease, but it cannot cure. The motor symptoms of Parkinson result from the death of the neuron cell in the substantia nigra. You know, when we talk about the neurons, some people say we have about 86 billion neurons in mind. Remember that. And none of the neurons are connected to each other. It is connected by synapse. So you can think how many things are there. So this is why to develop something for brain disease is really challenging, which is located in the midbrain and it causes a deficiency of dopamine. So जब आपके brain में dopamine का production कम होता है, उस समय dopamine neuron की death हो जाती है and Parkinson disease शुरू होता है. I will skip this. Let me just, okay. So if you want to work on this Parkinson, there is one target that is called NER1 enzyme. And NER1 enzyme was treated to be as an orphan receptor. Orphan receptor उसको बोलते हैं जिसका कोई binding pocket नहीं होता. But Professor Kim showed it has a binding pocket, but it is very tightly occupied. There is some mystery molecule present in our brain which binds with this NER1 and NER1 get activated. When it get activated, what it does, it stops the death of dopamine neurons. So when the death of dopamine neurons is stopped, the dopamine release increases, and that helps the treatment of this disease. So we worked on this concept. I'll skip this. So in a healthy humans, this is what the dopamine is. But if you have Parkinson's disease, the dopamine concentration decreases, and that leads to Parkinson's symptoms. So we started collaborating with him in 2012. Remember that in 2012. The first lot were 120 compounds, but based on what we had in our kitty, screened and proved that some of the compounds showed good activity profile, not very good. But later on, this was funded by MJ Fox Foundation, and we did extensive studies on that. Now, the EC50 value of chloroquine is 50 micromolar, which nature concluded it can use for the retardation of Parkinson's disease. Look at our molecules. Some of the compounds were active at the level of 0.5 nanomolar. These are additional compounds. So what we proved that T half of our compound is similar to chloroquine, number one. It has 75% better tissue penetration than chloroquine, and it has 74% better clearance than chloroquine. So we have US patent, PCT, then European patent, and 5 of those compounds entered to clinical trials. Let me just show you a couple of slides. What is the difference between this? What it was done? We say it is a NER1 enzyme है जिसके साथ वो bind करता है. ये हम दिखाना चाहते हैं. So what we did, it was done that the ligand bind domain of that enzyme was taken, and the assay was done with chloroquine and with our compound. For chloroquine, the activation of this NER1, the concentration of chloroquine was 100 micromolar, and our compound's concentration required only 20 more, 5 times better than chloroquine. So that was the ligand bind domain. Then whole enzyme was taken. Let us see what the effect is there. Similar kind. 5 times better. Then mutant neuron was taken, similar kind of result were obtained. And this shows the regain of the tissues after treatment with our compound. This shows the motor abilities of the mice improves when the dose level of 5 milligram is given to the mice. And this is a beautiful picture actually. This is the brain, cerebrum part of the brain of a mice. What was done? The experiment was done like this. The mice was injected a chemical molecule that what we call MPTP. The MPTP is nothing a chemical compound that causes Parkinson's disease. So Parkinson was induced. Now you can see the picture of the brain of a mice, very dull color. It was treated with L-DOPA, which is currently used to manage the Parkinson, improved slightly. Chloroquine, higher dose, regained little bit. Our compound 5 milligram per kg body weight regained the brain structure identical to the vacuum. So this is how we prove that this compound can cure the Parkinson's disease. Body weight improves during the treatment, after the treatment. All the pharmacokinetic parameters are equally good or better than chloroquine. And this was highlighted by media. And this is what we got at the end. Started with malaria, got many hit molecules there. And at the end, we have a molecule for the clinical trials that can treat the Parkinson's disease. And this is the website of the company called Norman Pharmaceutical. This is in Boston, in the United States, where our 3 compounds are listed in their website as a lead molecule and a molecule in pipeline. So one of the molecule is very close to clear the preclinical trial. After that, it will enter to phase 1, phase 2, and phase 3. Nobody knows what will happen. But we started getting the royalty now. The university has got the first part of the royalty just 2-3 months back. We work on other projects also, the catalysis. We have made a significant contribution in that area also. You can see the publications, and many of those papers has appeared in the cover page, highlighted by Synfact. And this all was possible because I was lucky to have very bright students working with me. So all of the students are well settled at their respective places now. And this is the current group of students who are working with me. And this is the group photograph was taken in 2020 actually at Indian Science Congress, where I was sectional president. And this is for the young students. Look, I had a broken leg. I had a plaster. I traveled all the way to Bangalore and conducted this 4 or 5 days program in different city. So you need to be very determined. But I showed you the success stories only. I have not shown the agony behind this story. That is much more difficult to explain at times. University of Delhi did not allow me to file the patent in the beginning. They asked me to pay from my own pocket in 2012 to file a patent. I did that. Funding agencies has some adverse comments on my project. So if you are determined, if you are good, if you have faith on yourself, you will succeed one day. And this is what the key of this is. With this, I thank each one of you for your patience here. Thank you very much.
Thank you, sir. Sir, today we are fortunate to hear about your research and achievements. And I'm sure this motivating and informative lecture is going to encourage our students and faculty members to pursue their research work in this area. Thank you, sir. Now I invite Professor Ruby Mishra to give a formal vote of thanks. Over to you, Ruby.
Rawat sir, you are requested to please stop sharing your screen.
Yeah, okay, okay. Yes. Yeah, thank you.
Will you please unmute yourself? We are not audible. We. Ruby says, please unmute her. Lina, Ruby is saying, please unmute her.
And she is the co-host. She can unmute.
But she's writing on the chat now. Unmute me.
Am I audible now?
Yes, Ruby.
Okay, I have joined from my mobile phone. There was some technical issues. So what a mesmerizing lecture by Professor Rawat. So thank you. We have come to the end of this technical session. So I would like to thank Dr. Roshni for introducing the keynote speaker of the session, Professor Rawat. And I'm very thankful to Professor Rawat for giving us the insight about the research work carried out in the Department of Chemistry under his supervision. And I'm really very happy to see the legacy of Department of Chemistry being carried out in such a great way, leading from Professor Sheshadri to Professor Parthasarthi, Professor Krishnamurti, Professor Khurana, who happened to be our teacher, and now you are leading the department and the legacy to such a grand level. And I'm thankful to my department and the conveners of the session, Professor Sushila and Professor Dr. Umesh Kumar, for giving me the opportunity to chair this session. I'm a very honored to listen to, fortunately, the lecture of Professor Rawat for the first time, and starting from the history of drug discovery, and then how that from a chemist and such a renowned chemist elaborating about the history of drug discovery way back from 1000 to 500 BC, and authenticating it, the Charak Samhita. And then what we know that in 1930s, the FDA was marked. So how rich was our Indian science, and especially our Indian or organic chemistry? So it was a quite interesting lecture, sir. Then he talked about the traditional drug discovery. And again, he brought about the concept of very new concept, because we have been teaching the undergraduate courses. So here the new thing which we knew, and it was really delight that our UG students have also joined this session, and they must now have got into touch of this concept of hybrid drug. So after going through this title of this talk, even I was finding it like how the anti-malarial and the Parkinson's was ये दोनों कैसे एक साथ जुड़े हुए हैं. And sir very beautifully explained about the discovery. तो हमें पढ़ते चलना चाहिए, चलते चलना चाहिए, पता नहीं कब क्या कैसे किस दिशा में चले और कहाँ क्या डिस्कवरी आ जाए. So great learning experience from sir Professor Rawat कि चलते चले, और we never know कि कहाँ हमें किस दिशा में. So determination and hard work is the key word which we can take from Professor Rawat. I'm thankful to Dr. Shilpi Bhatnagar for moderating this session, and also thankful to Dr. Leena Agrawal for handling the technical session of this international conference. So that's it from Dr. Shilpi. That's it from my side, Dr. Shilpi. Thank you. Over to you.
Thank you, Ruby. Now I request Dr. Leena to present a token of appreciation to Dr. Rawat. Thank you, Dr. Leena. Now I request Dr. Leena to present a token of appreciation to our chairpersons, Professor Ruby Mishra and Dr. Roshni Rajamohan.
This is very innovative way of presenting the memento actually. I saw this first time. Very good.
Leena,
Thank
please
you,
share
sir.
the screen again for sir. And Dr. Roshni. And सर का भी एक बार फिर से दिखा लेना. Now, please once again share Rawat sir token of appreciation and the e-memento. Alina? Okay, I think she's unable to hear. Okay. Thank you, Leena. Thank you,
Sorry,
everyone.
ma'am. I'm just
Acha. Okay.
Very good.
सर ये छोटा सा टोकन ऑफ अप्रिशिएशन है हमारे डिपार्टमेंट की ओर से.
Thank you, but
Certificate.
it's a
Thank you,
good
sir.
way actually. I saw this first time. कि ऐसा भी कर सकते हैं. Good, very good. Appreciate.
Thank you, sir. Thank you very much.
We will
Thank you,
present the hard
everyone.
copy also, sir.
हाँ,
We will present the hard copy also, sir.
अरे
हाँ.
अच्छा. Thank you.
Thank you, everyone, for joining this technical session. And now we approach to the valedictory session of the two-day international e-conference on recent advancements in chemical sciences, health, environment, and society under the aegis of IQAC and DBT Start College Scheme, organized by the Department of Chemistry, Deshbandhu College, University of Delhi. I, Dr. Shilpi Bhatnagar, would like to again welcome all the students, participants, faculty members, research scholars from India and abroad. With great pride and honor, I would like to welcome our chief guest, Professor Shri Prakash Singh, Director, South Campus, University of Delhi. Our guest of honor, Professor D.S. Rawat, Dean Examination, University of Delhi. Our principal sir, Professor Rajiv Agarwal, and the conveners of conference, Professor Shushila Singhal and Dr. Umesh Kumar. Now I invite Professor Rajiv Agarwal sir, Principal Deshbandhu College, to formally welcome our guest.
Thank you, Dr. Shilpi Bhatnagar. Since I am traveling, there are network issues, so I have not switched on my video. So, chief guest for the session, our very dynamic, humble Professor Shri Prakash Singh, Director, South Campus, University of Delhi. Very dynamic and scholar, Professor Diwan Singh Rawat, Dean Examination, University of Delhi. All the faculty members of Deshbandhu College and from other institutes and participants across the globe, Namaskar and a warm welcome to you all today as the Department of Chemistry is concluding a two-day-long international e-conference on recent advances in chemical sciences. I express my deep sense of gratitude to chief guest of the session, Director, South Campus, Professor Shri Prakash Singh, and Dean Examination, Professor Rawat. Both are very workaholic, yet focused to spare time for us, despite their extremely busy schedule. I would also like to thank our inaugural distinguished guest of the occasion, Professor Balram Pani, Dean of Colleges, University of Delhi, Professor A.K. Prasad, Head, Department of Chemistry, and Dean of Sciences, Delhi University, Dr. S.K. Vaishnav, Advisor and Head, International Cooperation, Department of Science and Technology, Government of India, who graced the occasion with their august presence. I would like to thank our governing body chairman, Shri R.N. Vats. I would take the opportunity here to thank all the participants, speakers, researchers, educators, and students who were present for the inaugural session as well as were engaged for the 2-day-long this e-conference in different capacities. Friends, chemical sciences have been playing an important role in developed and developing world, namely in the field of energy, water, agriculture, health, and many more. These areas need new materials and specific properties which chemical sciences satisfy. Chemical science forms a base for nanotechnology, providing polymers, clusters, and nanoparticles. Although the application of nanotechnology is gaining popularity, environmentalists have warned about the health hazard of nanoparticles. Significant advances have been made in harnessing energy from biomass and solid waste. The waste after gasification may be used as useful purposes. Use of biodiesel is an energy saver and pollution reducer too. This is being produced from Jaipropa, which grows in wasteland, needs very little water. The sun provides enormous energy to the earth, which has been converted to the thermal energy. I'm sure this 2-day of academic engagement will help us to find better equipped to deal in newer scope of scientific exploration. These are the snippets of the vast body of knowledge shared that I could gather so far. I hope all of you have been immensely benefited out of this conference. In the end, I would like to thank all the organizers and congratulate them, specifically the conveners of this program, Professor Sushila Singhal, Dr. Umesh Kumar, teacher in charge, Dr. Devnandan Kumar, and all the faculty members and students involving in this conference. I seek your presence and cooperation again as we continue to hold such conferences for days to come. It would be a wonderful academic journey together for the excellence of our nation. Thank you very much. Over to you, Shilpi.
Thank you, sir. Now I invite our chief guest, Professor Shri Prakash Singh, Director, South Campus, University of Delhi, to address the gathering. Over to you, sir.
Thank you so much. I'm audible properly?
Yes, sir.
Thank you. Thank you, Rajiv ji, having me here. A person from social science, political science primarily, is addressing scholars of sciences and the natural sciences, that is chemistry. And I was listening Professor Rawat, his journey, his efforts, his achievements, and the kind of deliberations he had with the scholars. That was amazing. I congratulate you, Professor Rawat, for making this event of this Bandhu College much more meaningful. I congratulate Ruby Misra, Devnandan ji, and other faculty members for putting an effort in this great endeavor of organizing such a program at college level and electronic mode, online mode, and having such a 172 participant at this juncture is another proof of success of this seminar. And what about chemistry? I know nothing. But I know this fact very well, that whatever we have in our society, even in our organic structure, functions on the basis of chemical reactions. our thought process, our happiness, our sorrow, each and everything is dependent on some chemical locha of our own organic structure. So chemistry is part of our life. We cannot think even an iota of our things without chemistry. दो इंसान बहुत अच्छे से बात कर रहे होते हैं तो हम कहते हैं बड़ी खूबसूरत केमिस्ट्री है। और जब मैं रावत साहब को सुन रहा था और अभी मैंने राजीव साहब को सुना तो झटरोपा प्लांट का जिक्र आया। रावत साहब, हमने 2002 में झटरोपा प्लांट पे दिल्ली में 2 बड़े कार्यक्रम किए। उन दिनों मैं कॉलेज में पढ़ाता था और एक वॉलेंट्री एफर्ट से हम लोग जुड़े हुए थे और उस वक्त प्लानिंग कमीशन के मेंबर थे डीएन तिवारी साहब। उनके मेजर 2 प्रोजेक्ट आए थे, एक प्रोजेक्ट झटरोपा पर था, एक प्रोजेक्ट जो था वो बैम्बूज पर था। आप सोच करके देखें कि फ्रॉम 2002 गवर्नमेंट ऑफ इंडिया हैव बीन वर्किंग ऑन दिस फील्ड, बट वेयर वी आर because of our lack of awareness, because of our somewhere economic and political interest in between, we have not moved much. But definitely I can say with my own experiences and exercise what we did in 2002 to 2005 pertaining to Jhatropa, that is an alternative viable for the energy. If we academics and our society move further for restoring this our ancient wisdom, even then that will help immensely. एक और छोटी सी घटना सुनाता हूँ, रावत साहब आपको रिलेट करेंगे या जो लोग इस वक्त जुड़े हैं वो शायद रिलेट कर पाए, रूबी भी शायद रिलेट कर पाए। रूबी की भी पृष्ठभूमि गांव की पृष्ठभूमि है। हम लोग जब गांव में पढ़ते थे तो हमारी पढ़ाई लिखाई केरोसिन ऑयल पर यानी मिट्टी के तेल पर डिपेंड करती थी। केरोसिन ऑयल नहीं मिला तो दिया जलना बंद, पढ़ाई बंद। का एक अल्टरनेट गांव देहात के लोगों ने खोजा, नीम का तेल। दिल्ली में तो सिर्फ टूथपेस्ट मिलता है लेकिन नीम का तेल भी मिलता है। और हम तो दिल्ली वाले लोग सिर्फ आलमंड ऑयल पानी ड्रॉप्स में पीते हैं। वो तो गांव देहात में कहा जाता है कि नीम की पत्ती खा लो तो आप बहुत सारी बीमारियों से दूर हो जाओगे। नीम का तेल जो है वो मिक्स करके पानी के साथ पी लो तो आपकी बहुत सारी दिक्कतें दूर हो जाएंगी। और ये जो सेप्टिक्स होते हैं, एंटीसेप्टिक्स होते हैं, तो शहर में मिलते हैं, गांव में तो मिलते नहीं, तो करके नहाते भी हैं। तो दूसरा हमारे पास आधार था नीम के तेल का, जिसपे दिए जलते थे। और जब नीम के तेल, क्योंकि बहुत ज्यादा रिस्टोर नहीं किया जाता था ये, तब रावत साहब अरंडी का एक पेड़ होता है जो झटरोपा के ही फैमिली का एक पेड़ माना जा सकता है, तो अरंडी का तेल जलाया जाता था। और मैंने ये स्थितियां स्वयं की हुई हैं, उनका अनुभव लिया हुआ, तो मैं कह सकता हूं कि जो हमारा एंशिएंट नॉलेज सिस्टम था उसको हमने बहुत महत्व नहीं दिया, उसको ट्रेडिशनल नॉलेज कह के, रूरल इंडिया बेस्ड नॉलेज कह करके हमने उसकी उपेक्षा कर दी। एक और छोटी सी कहानी मुझे बड़ी प्रिय है, इस नाते सुनाता हूं। अब लोग सोच रहे होंगे कि केमिस्ट्री की क्लास है, केमिस्ट्री के स्कॉलर्स हैं, सोशल साइंस वाले को बुला लिया, वो कहानी सुना रहा है। लेकिन बुलाया है तो थोड़ी देर आप झेलो मुझको। मैंने कल्याण, एक गोरखपुर प्रेस जो गीता प्रेस है, वहां से एक मैगजीन निकलती कल्याण, and it's a beautiful narration of how our nature is full of chemicals. And in that story, A and B both are Ayurveda Tarats. A invites disciple and asks for to B. But while going to B, my advice is to, when you eat, when you stay, when you sleep, be with the bubbles tree. वो कह रहा सिर्फ बबूल के पेड़ के साथ आप रहिए, उसके नीचे खाइए, उसके नीचे सोइए, उसके नीचे विश्राम करिए. Disciple follows the order of Guru, and when he reaches to the B, the second Ved Visarad, by that time his whole skin was full of skin disease. And finally, he gives a message to B and says that this is a message by my Guruji to you. उसमें लिखा क्या हुआ था कि आप बहुत बड़े वैद्य हैं, अब इसको ठीक करके बताइए. उन्होंने इसको पढ़ा, मुस्कुराए, जेब में रखा, बोले भाई देखो वो अरे गुरुजी बहुत बड़े आदमी हैं, ठीक तो तुम्हें वही कर सकते, मैं तो कर नहीं सकता. लेकिन मेरी एक सलाह है, अब जब तुम वापस जाना तो तुम नीम का उपयोग अपने जीवन में करना. नीम के नीचे सोना, नीम के नीचे खाना, और सुबह उठ करके 2 पत्ती नीम की जरूर, और जब स्नान करो तो नीम की पत्ती के पानी से करना. अब वो बेचारा कर जब वो पहुंचा, पहले से ज्यादा हैंडसम था, पहले से ज्यादा स्वस्थ था. अब आप सोच रहे होंगे मैंने कहानी क्यों सुनाई. अ केमिकल रिएक्शन ऑफ बबूल is much more futile to the बंजर लैंड, बारन लैंड जिसको हम बोलते हैं. वहां पर उसकी प्रोडक्टिविटी रिस्टोर करने में सिर्फ वही पौधा काम आता है, कोई दूसरा पौधा काम ही नहीं आता है. और अभी आप जो लोग गांव समाज से जुड़े, जिनका बचपन या आज भी जिनका जीवन गांव से जुड़ा हुआ होगा, तो हर दरवाजे पे नीम लगा मिलता है. नीम के नीचे लोग बैठते हैं, नीम के नीचे लोग सोते हैं. अब जरा अर्बन लाइफ की बात कर लीजिए. Around 65% of our population still lives in villages, and majority of their lives and concerns is associated with such trees which have immensely medicinal values for the life. तो प्रकृति तो हमको रोज बताती है कि आई एम फुल ऑफ केमिकल, फुल ऑफ केमिस्ट्री, but it is you and me who have to make our relations with that. अपना केमिकल रिएक्शन एंड केमिकल केमिस्ट्री जो है उनके साथ ठीक रखना है, इसकी शिक्षा हमको रोज प्रकृति देती है. अब जरा हम एटम्स की बात कर लें. एटम्स के बारे में कहा जाता है कि 2500 वर्ष पहले ऋषि कणाद ने इसके कुछ सूत्र दिए थे. जब एटम बना तो उसके ऊपर जे रॉबर्ट ओपेनहाइमर रिलेवेंस ऑफ भगवत गीता एंड इट्स इन फॉर्म ऑफ सूत्र एंड एसोसिएट्स इट्स सिग्निफिकेंस. यानी ओपेनहाइमर कहता है कि गीता में इसके सूत्र हमको मिलते हैं. लेकिन हमारा दुर्भाग्य है कि जब हम एकेडमिक्स में भगवत गीता की बात करेंगे तो कुछ लोगों को दिक्कत होने, उनका केमिकल रिएक्शन कुछ कुछ होने लगता है. और वो गीता जो कम्युनल हो जाती है. ओपेनहाइमर कहता है कि गीता में एटॉमिक रिसोर्सेज हैं, नॉलेज है, विजडम है, पीस है, प्रॉस्पेरिटी है, डिप्लोमेसी है, और हम उसमें कुछ कुछ खोजने लगते हैं। इस मानस से भी ऊपर उतरना चाहिए। कभी हमने सोचा है क्या कि साउथ इंडिया हम जाते हैं, तमाम बड़े बड़े मंदिर हमको देखने को मिलते हैं, वो ग्रेनाइट वहां पैदा नहीं होता, उतने बड़े हॉल्स वहां पैदा नहीं होते, वो पत्थर भी वहां पैदा नहीं होता। तो क्या किसी बगैर टेक्नोलॉजी के सब कुछ पहुंच गया? इतने बड़े बड़े मंदिर बना दिए। उत्तर भारत में तो इतने बड़े मंदिर है ही नहीं। मीनाक्षीपुरम का टेम्पल देखिए, वो रॉक्स तो वहां मिलती ही नहीं है। तो बगैर टेक्नोलॉजी के बना क्या? यानी टेक्नोलॉजी थी, उसके यूसेस थे। 1757 तक हम वर्ल्ड इकोनॉमी का 26 to 28% कंट्रोल करते थे। जब ब्रिटेन इंडिया आया तो उसकी इकोनॉमी क्या थी जीडीपी में? 1.2%। जब गया तो उतने पे हमको देखे गया। स्पेन एंड पुर्तगाल both decided to have the petition for India. What was their economic contribution? Minimal to the world economy. They wanted to have a trading ties with India through a shortest route. That was Columbus and Vasco da Gama. तो इतनी इकोनॉमिक प्रॉस्पेरिटी ऐसे तो आई नहीं। हमको साइंस का भी ज्ञान था, तकनीक का भी ज्ञान था, और बाजार का भी ज्ञान था। जरा चोलाज के, राजेंद्र चोला के बारे में गूगल करिए, पढ़िए। सुमात्रा भी हमारा था, मलाया भी हमारा था, और वियतनाम में तो मैं खुद देख करके आया हूं शिव के मंदिर और सारी मूर्तियां। Rawat Sahab, you will be surprised to know a lingam where all three gods, Vishnu, Shiva and Brahma, in Vietnam. It's a communist country, but their maximum revenue is coming from two places. One is associated with Gautam Buddha, another is associated with Rajendra Chola's activities in 2nd BC, and that is of Hindu religious places and Hindu deities. तो मेरा तो सिर्फ मित्रों से आग्रह, जरा खोज करके देखो। बहुत ज्यादा अध्ययन करने की जरूरत भी नहीं है, सिर्फ आइडेंटिफाई करके गूगल बाबा की मदद ले दो, वो भी बता। अब मैं आता हूं, थोड़ा सा मैं अपनी बात समाप्त करता हूं, क्योंकि रावत साहब को भी कुछ बोलना। एक और बात, कौटिल्य का नाम हम सबने सुना है। कौटिल्य का जो 15वां चैप्टर अर्थशास्त्र का, उसको बोलते हैं तंत्र युक्ति। एंड तंत्र युक्ति talks about 30 research, and you will be surprised, in this age of scienticization we may not remain neutral. Our researches are 99% are tilted towards A to B or C, but there in Tantra Yukti, Kautilya tells about how to maintain neutrality while writing, while researching. रावत साहब, उसको कभी पढ़ने का अवसर लगेगा तो आपको आनंद आएगा। कौटिल्य कहता है, हेतुवर्ताः, हम रिसर्च क्यों कर रहे हैं, हेतु क्या है इसका, हु विल फंड इट, एकांत नैकांत, यानी रिसर्च जो भीड़ में नहीं होती है, एकांत में होती है। पूर्व पक्ष, अपपक्ष, ये सारे 32 सूत्र देता है, और साथ में लिखता है इन सुश्रुत there are 36 tools. सुश्रुत कौन है हमको बताने की जरूरत नहीं। हमारे यहाँ जो सर्जरी की शुरुआत है उसके फादर माने जाते हैं। हम तो शायद उतना सम्मान नहीं देते, लेकिन विदेशों में बहुत सम्मान है। सुश्रुत have been using in his research 36 tools. आप सभी तो साइंस के विद्यार्थी हैं, जरा सुश्रुत के टूल्स, रिसर्च के टूल्स खोज करके देखिए, वो 36 टूल्स क्या हैं। and apply those tools to your modern researches, you will find that perhaps Susrusto much more systematic. क्योंकि हजारों साल पहले उन्होंने कुछ इन्वेंट किया, कुछ बात की, तो उसको उपेक्षित कर दिया जाए इन फॉर्म ऑफ मॉडर्न साइंस, वो कहीं से भी विज्ञान के साथ, मानवता के साथ उचित नहीं। और अंत में मैं ये कहना चाहूंगा कि कम से कम जो रचनाएं हमारे विद्वानों ने की हैं, आचार्य प्रफुल्ल चंद्र रे has written 2 volumes on history of Hindu chemistry, ancient to medieval. एक उन्होंने 1902 में एक वॉल्यूम लिखा, दूसरा 1907 में लिखा। तो हमारे केमिस्ट्री के साथियों को एक बार तो जरूर पढ़ना चाहिए, और जब वो पढ़ लेंगे तो मुझे लगता है कि बहुत लाभ उनको भी और हमारी जनरेशंस को होने वाला है। अंत में अपनी बात समाप्त करता हूं ये कहते हुए कि राजीव जी ने, रूबी मिश्रा जी ने, देवनंदन जी ने, उमेश जी ने, आदित्य ने, पूरी टीम मुझे जो दिख रहे हैं नाम, शिल्पी भटनागर जी ने, आप सब ने एक बहुत अच्छा उपक्रम किया। साइंस जो है, विज्ञान जो है, हम सबकी जरूरत भी है, लेकिन उसका मानवीय मूल्य सदैव बरकरार रहे, इसका हम सबको ध्यान रखना चाहिए। बहुत बहुत साधुवाद, बहुत बहुत धन्यवाद।
Thank you, sir, for your inspiring words and sharing your thought-provoking experiences. Thank you once again, sir, for sparing your valuable time. Now, I request Dr. Leena to present a token of appreciation and e-memento to our chief guest, Professor Shri Prakash Singh. To Leena. Now I would like to invite our guest of honor, Professor D.S. Rawat, Dean Examination, University of Delhi, to address the gathering. Over to you, sir.
Yeah, thank you. Professor Shri Prakash Singh ji, Dr. Rajiv Agrawal, Dr. Ruby, Shilpi, Dr. Umesh, other colleagues. Let me first congratulate to all of you for organizing such a wonderful program. I was going through the list of speakers. Actually, in scientific deliberations, the most important part is the kind of speakers you invite. That sets the tone and success of the conference. In that token, I'm really, really pleased that you could invite, arrange their talks of some of the stalwarts in the field. So congratulations to all of you. Otherwise, in most of the places, what happens, we just organize a talk and then, the speakers are not of that quality. So I must congratulate all of you for this event. Professor Shri Prakash touched upon many areas. Actually, he did not left anything for me to speak about. And it was really amazing to see that being a social science person, he spoke and touched upon almost all areas of chemistry. And at the end, he even talked about the Professor P.C. Ray's, the classical book, that is History of Hindu Chemistry. Unfortunately, he was lucky actually at that time, at least in 1902 and 1907, nobody termed him as a communal, actually, using this term. And the people even don't know actually these 2 books were reviewed by Nature, and Nature wrote a review article on the book which Professor P.C. Ray published it. I wanted to purchase this book actually a couple of years back, and it was out of stock, right? And I compliment the government, actually they made an effort and the book is now available. I could purchase just 2 years back now. And it's wonderful text to read. And this is what we are actually. We are not the only that what we have been taught since our childhood, unfortunately. We are not connected to our roots. We are connected to, in between somewhere. I'm a science student, so I don't know much of history in that way, which the social science and the historian knows. But yes, wherever I visit, I just think about the aspect what Professor Sri Prakash said. Look at those temples. Even if you are, अगर आप नास्तिक भी हैं, तब भी जाकर के आप देखिए, सोचिए, 1000 साल पहले जब उसको बनाया होगा, उसमें कौन सी टेक्नोलॉजी रही हो. So just think about it. This is what it is. I happened to be once to in Rajasthan. Actually, there is one temple. I was told कि सास बहू का temple. So I was more curious. जब सास बहू temple का, मैं इसको तो देखना ही थी. So वहां गए तो पता चला वो सहस्त्रबाहु temple. And सहस्त्रबाहु temple was archaeological survey of India ने 1955 या 56 में उसको निकाला क्योंकि वो पूरा दबा हुआ था. And the history says that temple was ruined by Iltutmish, maybe 1100 or 1200 years. And after that, we have so many rulers, and even we term them as a Mahan, or the great, but they did not restore our dignity. This is what our culture was. So when we talk about all that, we should not forget the role of the nature. Our Vedas were much more competent than our surgeons of today. Actually, if you visit to any of the doctor today, they will not give you anything unless all the physiological tests are done. Talk your grandfather. Father को भी आप शायद पता न हो, गांव में जो रहते हैं उनको तो पता होता है. Our Vaidya's used to detect your diseases by looking at your pulses. अगर आप याद हो उन लोगों को जिनने गांव की जिंदगी देखी है, तो वेदास के लिए वो कुछ करने की जरूरत नहीं होती, उनके पास कोई आला नहीं होता था. वो आपकी नाड़ी पकड़ते थे, 10-20 मिनट के लिए नाड़ी देखते थे और आपको बताते थे ये है और ये लेना है, और उससे ठीक होते थे. वो मैंने अपने आंखों से भी देखा. पहाड़ों में तो हमारे यहाँ एक्चुअली में नीम वगैरह कम होता है ऊपर हाई एल्टीट्यूड में, तो हमारे यहाँ केरोसिन के बाद चीड़ होता है. एक्चुअली चीड़ का ट्री होता है, तो चीड़ का ट्री में रेजिन बहुत ज्यादा होता है. तो रेजिन वाला जो पार्ट था उसको हम लेते थे और उसको जला करके लाइट करना या कहीं जाना है उसको जाकर के. रेजिन इज यूज फॉर मेनी पर्पसेस. सो द किड्स नीड टू बी टॉट अबाउट दोज एस्पेक्ट्स जो हमारे उसमें था. एनसीईआरटी टेक्स्ट बुक्स में हम लोग कुछ उसमें डाल देते हैं टेक्स्ट बॉक्स आइटम्स में, एंड उसमें भी एक बार बड़ी मुश्किल हो गई थी जब उसमें ये कहा गया था कि sun is considered to be source of energy as per our traditional books or the Vedas, and there was a difficulty to include that in the textbook. Actually, it was removed at the end. So my message to all of the students is this subject is really fascinating. Okay, so do hard work, and all the faculty members, I request all of you to contribute to make the quality work. Do only one small thing, and that should be your quality. Okay, that will help the university and the college to grow beyond what we are today. Thank you very much. Thanks for listening. Thank you.
Thank you, sir, for your inspiring words and constant support and encouragement to all of us. Now I request Dr. Leena to present a token of appreciation and e-memento to our guest of honor, Professor D.S. Rawat. Thank you, Leena. Now let's proceed further. The wait is over, and now it's time for the declaration of results. I would like to invite our faculty members of Department of Chemistry, Deshbandhu College, Dr. Shilpi Khurana and Dr. Ishwar Dutt to announce the results of oral presentation and poster presentation. Over to you, Dr. Shilpi and Dr. Ishwar.
Good afternoon, everyone. Am I audible to everyone?
Yes, you are.
Good afternoon, everyone. I'm here to present the 5 best presenters in oral presentation. Number 1, OP36, Dr. Elena Gauri, Department of Ras Shastra and Bhagyasya Kalpana from All India Institute of Ayurveda, New Delhi. Number 2, oral presentation number 41, Dr. Om Prakash, Department of Chemistry, University of Copenhagen, Denmark. Number 3, OP47, Mr. Keshav Kumar Saini, Department of Chemistry, University of Delhi. At number position 4, we have OP140, Ms. Dipali Aluwalia from Delhi Technological University. At position 5, we have 2 oral presentations selected by the members. We have OP55, Ms. Priya Jain, Department of Chemistry, IIT Delhi. And at the same position, we have OP150, Dr. Deepak Gupta, Catholic University of Catholic Belgium. So these are the 5 best presenters in the oral presentations category. Many congratulations to everyone. Over to you, Dr. Shilpi Bhatnagar.
Dr. Ishwar, are you available? Please announce the results for poster presentation as well. Dr. Ishwar.
Very good evening to all of you. Am I audible?
Yes, you are.
Okay. A very good evening to our chief guest, Professor Shri Prakash sir, and our guest of honor, Professor D.S. Rawat, and to our own principal sir, Professor Rajiv Agarwal. So it is indeed a privilege for me to announce the result of the poster presentation. But before announcing the result, I would like to inform that we have received large number of posters, and approximately 65 posters were there, and 4 simultaneous sessions were there for the poster presentation. And posters were judged on various parameters by 3 independent judges for each group. And it was very difficult to select few posters because all of the posters were very good and the presentation was very nice. But since we have to do this work, and I'm here to announce the result. The results are not in any chronological order. Among the various posters, we have selected 5 best posters. So again, I'm saying that they are not in any order. We have selected just 5 best posters. So first poster, which is among the best poster, is poster number 14, which is presented by Meetu Luthra from the Department of Physics, Bhaskaracharya College of Applied Science, University of Delhi. The second poster among the best is poster number 16, presented by Anu Manas, Department of Chemistry, School of Technology, Pandit Deen Dayal Energy University, Gandhinagar, Gujarat. Then the next poster is poster number 36, presented by Sohan Singh from Department of Chemistry, School of Chemical Sciences and Pharmacy, Central University of Rajasthan. The fourth poster among the best poster is poster number 50, which is Ashdeep Kaur Gill, presented by Ashdeep Kaur Gill from the Institute of Nanosciences and Technology, Sector 81, Knowledge City, Mohali. And the last poster among the best poster is poster number 57, which is presented by Akashdeep Singh Pathania from the Department of Pharmaceutical Sciences and Drug Research, Punjab University, Patiala. So many congratulations to all the winners of this Best Poster Award. Thank you so much for giving me this opportunity. Now over to you, Dr. Shilpi.
Thank you, Dr. Shilpi Khurana and Dr. Ishwar Dutt, and many congratulations to all the winners. An important announcement for everyone to fill the feedback form to get the e-certificates. Now, I would like to invite Professor Lieutenant Sushila Singhal to give a formal vote of thanks and to present the report of this 2 days international e-conference. Over to you, Sushila ma'am.
Thank you, Dr. Shilpi. Good afternoon, all. Honorable Chief Guest, Professor Shri Prakash Singh, Director, South Campus. Guest of Honor, Professor D.S. Rawat, Dean Examination, University of Delhi. Chief Patron, Shri R.N. Watts, Chairman, Governing Body, Deshbandhu College. Respected Principal, Professor Rajiv Agarwal, Deshbandhu College, and all participants. It's my honor and privilege to be convener of 2 days international conference entitled Recent Advancements in Chemical Sciences, Health, Environment, and Society, organized by Department of Chemistry, Deshbandhu College, and also to propose a formal vote of thanks on this occasion. I, Professor Sushila Singhal, on behalf of Department of Chemistry, Deshbandhu College, and organizing team, extend a very hearty vote of thanks to the honorable delegates, Chief Guest of inaugural session, Professor Balram Pani, guests of honor, Professor Ashok Kumar Prasad and Dr. Sanjeev Kumar Vashne, our Chief Patron, our Chairman, Governing Body, Deshbandhu College, Shri R.N. Watts Ji, Chief Guest of valedictory session, Professor Shri Prakash Singh, and Guest of Honor, Professor D.S. Rawat. Now, I would like to extend a special thanks to patron of conference, our principal, Professor Rajiv Agarwal, for providing encouragement and constant support at every moment to make program a success. Thank you, sir. Now, I wish to express my gratitude to coordinators of conference, Vice Principal Professor Kamal Kumar Gupta, IQAC Coordinator Dr. Aditya Saxena, DBT Star Skill Coordinator Dr. Indrakant Kumar Singh. I must mention our deep sense of appreciation to our dynamic teacher in charge, that is head of our department, Professor Devnandan Kumar, who wrapped all flowers of our department in a garland, which has been self-showcased by their coordination throughout the program. Professor Devnandan always put efforts for the betterment of department and college as well. Thank you, Professor Devnandan. As no program can become successful with a single person, so I extend my big thanks to our dedicating organizing team, our technical team, moderators and chairpersons of the sessions, working tirelessly from beginning to completion of the event. Thank you all. I must thank to advisory team of International Conference for providing valuable inputs in organizing events successfully. Since every program involves some monetary transactions and investments, I would like to thank sponsors of the conference, Punjab and Sindh Bank, States Hub Enterprises, and Jen Chemicals. My hearty congratulations to the winners of oral and poster presentation and all participants for their active participation in this event. Prior to winding it up, an announcement is there for our department upcoming conference, which is coming very soon under the convenership of Dr. Pratibha. I wish to see you all in upcoming event. Once again, thank you all for making this event successful with your contribution. Thank you. Now I would extend to the report of the conference. Report of 2 days conference, International E-Conference on Recent Advancements in Chemical Sciences, Health, Environment and Society under the aegis of IQAC and DBT Star College scheme included the inaugural session and valedictory session along with the 5 technical sessions. The inaugural ceremony of 2 days International E-Conference was held on the web platform Zoom on 8th and 9th April 2022 and hosted by the Department of Chemistry, Deshbandhu College, University of Delhi. The program was started by the moderator, Dr. Namita Gandhi, Associate Professor, Department of Chemistry. She welcomed Chief Guest of the program, Professor Balram Pani, Dean of Colleges, University of Delhi, and Chairman R.N. Watts, Chairman Governing Body, Deshbandhu College, and our 2 guest of honors, Professor A.K. Prasad and Dr. Sanjeev Kumar Vashne. Professor Kamal Kumar Gupta welcomed and also presented his sincere thanks to all participants and organizers. Dr. Umesh Kumar welcomed to all and introduced the objective of the conference. He said that all the major area of research related to chemical science, health, environment, and societies have been covered here. Total 166 oral and 63 poster presentations were received from graduate and postgraduate students, research scholars, and faculty members from India and also from abroad. Professor Balram Pani congratulated to Department of Chemistry and organizing committee members. He also said that chemistry is present everywhere in our society. He presented his best wishes for the success of this e-conference. Dr. S.K. Vaishnav congratulated to all. In his speech, he said that the existing technology in chemistry have a great significance for the development of environment and society. He included some beautiful words which shows the significance of chemistry, that life is a chemical reaction, just balance it. Professor Ashok Kumar Prasad congratulated teacher in charge, Professor Kumar Devnandan, convener and organizing committee for conducting such a wonderful e-conference, which based on many themes. E-abstract book of the conference was launched in virtual mode by the moderator. Professor Devnandan Kumar, teacher in charge, Department of Chemistry, welcomed all and talked about the significance of the conference and concluded remarks of the inaugural session. Professor Sushila Singhal, that is myself, presented a brief introduction about keynote speaker, Professor Maya Shankar Singh, Department of Chemistry, Banaras Hindu University. Professor Singh delivered a wonderful lecture and focused his talk on heterobonds, free radicals, pericyclic reactions, addition reactions, and polyfunctional molecules, and many more. After completion of the inaugural session, token of appreciation and e-mementos were presented to all delegates. And then we moved to the technical session 1. Moderator of this session was again Dr. Namita Gandhi, and chairpersons were Professor Devnandan Kumar and Professor Mahesh Chandra. In this session, an invited talk was delivered by Professor Praveen P. Ingole, Department of Chemistry, IIT Delhi. His lecture focused on the minimization of carbon dioxide gas and its conversion into other useful products by following the innovative techniques such as by use of copper amide as a catalyst. Second speaker of this session was Professor Utpal Bora, Department of Biosciences and Bioengineering, IIT Guwahati. He explained that free trades and globalization machine is connected with pollution and carbon dioxide emission, environmental destruction, consumer junk, global cheap labors. Token of appreciation were presented to both the speakers and chairpersons. Then we moved to the technical session 2, which was handled by Dr. Seema Lal, Assistant Professor, Department of Chemistry, Deshpande College, with a welcome note for invited speakers, participants, and organizing team, followed by a brief introduction of chairpersons of the session, Professor Surinder Singh and Dr. B.K. Singh from Department of Chemistry, University of Delhi. Chairperson Professor Surinder Singh has introduced and invited the first speaker, Professor Kasturi Mukhopadhyay from JNU, for her invited lecture. During her lecture, she emphasized on the working mechanism of antibiotics, timeline of discovery, and novel antibiotics. Professor Kasturi has highlighted the properties of curcumin, an Indian spice, as natural antimicrobial agent due to the presence of beta-diketone moiety, alkene link, and phenolic group in its structure. Next invited speaker, Professor Bhumi Shankar, IISER Pune, was invited by Dr. B.K. Singh with a brief introduction. Professor Bhumi Shankar discussed and concluded the development of non-crystalline polymer composites of ferroelectric salts and compared these composites for mechanical energy harvesting applications like capacitors, charging, and LED flash lighting applications. Session was concluded by Dr. Seema Lal with a thank note to both the invited speakers and chairpersons in the form of e-memento and token of appreciation. She also thanked organizing committee members and to all participants. She made an important announcement related to the upcoming oral and poster presentation sessions. After technical 2, we moved to oral and poster sessions, which were held simultaneously in different groups at Google Meet. Participants actively participated in both sessions. And with this, the day 1 was over. Then we moved to the day 2 in the technical session 3, which was moderated by Dr. Parul Singh. She welcomed the chairperson, Dr. Aditi Saxena and Dr. Aparna Shekhar. Dr. Aditi Saxena welcomed Professor P.S. Mukherjee, who talked on the molecular vessels. Dr. Aparna Shekhar, she invited Professor Amitabh Jha, who discussed on the novel selective estrogen receptor modulators. After their session, Dr. Parul Singh, she presented the e-memento and token of appreciation to both the speakers as well as to both the chairpersons. And we moved to technical session 4, which was again moderated by Dr. Parul Singh and chairpersons Dr. Manisha Sengar and Dr. Indrakant Kumar Singh. Dr. Manisha Sengar, she welcomed Professor Niranjan Parajuli, who discussed on the genetic characterization of actinomycetes useful for the antibiotic production. Dr. Indrakant Kumar Singh welcomed Dr. Rais Ahmed Khan, who has discussed and concluded about the human serum albumin binding affinity and anti-cancer potential of the bioactive organic moieties, suspended ruthenium complexes. After this, we moved to the technical session 4, which is moderated by Dr. Shilpi Bhatnagar. Chairpersons of this session was Professor Ruby Mishra and Dr. Roshni Raja Mohan. Dr. Roshni welcomed Professor D.S. Rawat sir, Dean Examination, University of Delhi, New Delhi, India. Professor Rawat has given a talk on journey from anti-malarial molecules to clinical candidate for Parkinson's treatment. After his talk, a token of appreciation and e-memento was presented to Professor Rawat, and we moved to the valedictory session. In the valedictory session, Dr. Shilpi Bhatnagar, she invited and welcomed our chief guest, Professor Shri Prakash Singh, and guest of honor, Professor D.S. Rawat. She also welcomed Professor Rajiv Agarwal, and he spared his valuable time, although he is in his journey, but he spared time and he welcomed our chief guest, Professor Shri Prakash Singh, as well as Professor D.S. Rawat. We announced the result of oral and poster presentation by Dr. Shilpi Khurana and Dr. Ishwar Dutt. Vote of thanks was presented by Professor Sushila Singhal, and she read the report also. Thank you all. Over to you, Dr. Shilpi.
Dr. Sushila. Now I request everyone to switch on your cameras for a group photograph. Please switch on your cameras. Selena, please do the needful. Selena, please do the needful.
Ma'am, I have given the permission. Now everybody can switch on their video. I have given the permissions. Everybody will be allowed. All the participants have been given permission for switching on their video, so they can switch on their video for the group picture. Now Ashwini sir is requested to please take a group photo, sir.
Yes, ma'am. Done, ma'am. Done.
Oh, done.
Thank you, everyone. Now let us all stand for the national anthem. I request the technical team to play the national anthem.
I request all of you to please stand. I'll give you some time and then I'll play. Please stand and adjust your videos.
जन गण मन अधिनायक जय हे भारत भाग्य विधाता। पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंगा। विंध्य हिमाचल यमुना गंगा उच्छल जलधि तरंगा। तव शुभ नामे जागे तव शुभ आशीष मांगे। गाहे तव जय गाथा जन गण मंगल दायक जय हे भारत भाग्य विधाता। जय हे जय हे जय हे जय 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 हे।
To everyone, I once again thank you all for your precious time and patience and making this 2 days international e-conference a huge success. Thank you, everyone. Many congratulations to all.
Should I end the meeting then?
Yes, I think. Feedback form is posted in